Well, thanks, Graham. Thanks to CPAF, and thanks especially to NCATS for the support for our work. And I'll talk today about the work we've been doing for an extended period of time with developing complex in vitro models for different kidney-related uses. And kidney diseases are a growing, I think it's fair to say, public health problem, affecting close to 15% of the US adult population. Almost between 10 and 14% of the global population are considered the 10th leading by the CDC, 10th leading cause of death in the US, but are projected to become the 5th leading cause of death around the world by the year 2040. So they're certainly increasing. The costs are enormous to the federal government and particularly to Medicare. But as a nephrologist, what's exciting is that for the first time, we've really had breakthroughs in the development of really blockbuster new therapeutics. The flozins, the SGLT2 inhibitors, the GLP-1 agonists now have been shown to slow the progression of kidney disease along with aldosterone antagonists. And I think because of this, as somebody who's been doing this for 35 odd years, there was very little progress in drug development in kidney disease and very little interest on the part of pharma and biotech in working on kidney diseases. But there's been an explosion of interest now in the last 5 to 10 years, both on the part of large pharma and on the part of biotech. And now that it looks tractable to come up with good therapeutics for kidney diseases. The flip side of developing new therapeutics is drug-induced kidney injury, which is actually quite common. Particularly the kidney has a very complex architecture, but the proximal tubule cell gets exposed to very high concentrations of drugs, either that are filtered through the glomerulus and then taken up apically into the cell, or that are secreted basolaterally through renal blood flow. Either way, the intracellular concentrations of this very metabolically active cell of many drugs and xenobiotics is very high. And really, the proximal tubule, which is one of the most metabolically active cells in the human body, the main work of the proximal tubule is transport, either reabsorbing or secreting various components of the circulation. Now, one distinction I want to make, and I think this is important, between nephrotoxicity or drug-induced kidney injury and, for example, DILI, which we've had a lot of focus on today, is that many drugs that are known to be nephrotoxic are still in the clinic and are still prescribed. And what's needed is recognition of the potential nephrotoxicity and then strategies to mitigate the risk. But it's not a death sentence for a drug to find out that it's nephrotoxic. And I'll show a few examples of that going forward. So our group, which has been working in this area now for over a decade, and we're really indebted to NCATS for having the vision to create these microphysiological systems. Our team's been together for an extended period of time, and we proposed 5 contexts of use for drug development tools as part of the TRACE MPS program. 2 are really on the tox side, and they're both related to proximal tubule function and really to our kidney on a chip or proximal tubule on a chip microphysiological system. One is a PBPK, physiologically based pharmacokinetic model that we've built also for how the kidney eliminates drugs. And then 2 are using iPS-derived kidney organoids for the drug development, drug discovery side, and for disease modeling. So it really covers the gamut in terms of development of drugs that affect the kidney. Now, this team, I really want to emphasize, I have been privileged for over 10 years now to work with the same individuals in this whole process. And it's really an amazing team, and they deserve all the credit for all the data that we've generated. Ying Zheng is an amazing biomedical engineer and vascular biologist. Ed Kelly is a toxicologist. Kathy Young is a PharmD PhD pharmacologist, and Bino Friedman is a expert in stem cell biology and organoid development. And together, this team has really led all of the work that we've done over this period of time. And I'll show you a few examples from work we've done actually often a while ago that are relevant to the 5 contexts of use. So this is a study that we published a decade ago where we took 39 compounds, and we did this in collaboration with Vishal Vaidya, who at the time was at Harvard and now he's at Pfizer. But we took 39 known nephrotoxic drugs and did both 2D and 3D in vitro systems to try to understand and predict the nephrotoxicity. The 2D system being more high throughput and the 3D so-called proximal tubular kidney on a chip for greater specificity. 4 different doses, 4 different readouts. And what we found is that if we added the 3D organ on a chip or proximal tubule on a chip to the 2D higher throughput toxicity assays, we could increase the AUC for detecting nephrotoxicity up to 0.92. So we only missed 2 out of the 39 drugs when we combine the organ on chip work with more high throughput 2D screening. And also, just to point out the drug here, and what we found is that upregulation of hemoxygenase 1 was a very potent signal of nephrotoxicity. We developed fluorescence assays for this, fluorescent reporter lines for this, for our chip work. And I point out that tacrolimus is a drug. You can see how much it upregulates hemoxygenase 1. It is a nephrotoxic drug, but the calcineurin antagonists like tacrolimus are used all the time as immunosuppressive drugs. It's the mainstay of drugs that are used now for immunosuppression still with solid organ transplantation. So again, just because a drug is nephrotoxic doesn't mean it won't get into the clinic and it won't be prescribed, but you have to understand it. You need to know about it a priori. You need to mitigate it and do risk-benefit kinds of analyses. Another drug that we studied, again, about 10 years ago was polymyxin B. Polymyxin B is an antibiotic that has been known and used for almost half a century now. It's a very nephrotoxic antibiotic. And I should mention that antibiotics and antivirals are often nephrotoxic drugs. But it's one of the most nephrotoxic drugs that you can prescribe. But it is used as a last resort because it's still a very good antibiotic when there's a lot of microbial resistance. So we studied this drug and we worked with a small company that was making congeners of this drug that they thought had the same antibiotic activity, but would be less nephrotoxic. These were a very small company of medicinal chemists. And this is a very polycationic drug, polymyxin B. They made it less cationic. Aminoglycosides, which are also nephrotoxic, are very polycationic as well. And what you see in red is upregulation of kidney injury molecule 1 with polymyxin B on chip, but the other 2 congeners, weren't upregulated. After that, after we did this study, it was taken into the clinic for a phase 1 trial, these 2 congeners, and were found not to be nephrotoxic. Then the small company sold them to somebody as an asset who decided not to develop it further. So this would have been a really nice example of chip work going into the clinic if only they had further gone into the clinic. KIM-1 we will be using as one of our biomarkers for toxicity in these systems, and it has the advantage that KIM-1 was qualified in 2010 by the FDA for animal model toxicity as being more sensitive and specific than a rise in serum creatinine, and then was qualified also as in the clinic as a biomarker of acute kidney injury. So there's a predicate for using KIM-1, and we've found it to be a very effective biomarker, as well as some of the microRNAs that you see over on the right-hand side. So those are sort of some of what we've been doing that's relevant to the 2 contexts of use for toxicity. We've also developed physiologically based pharmacokinetic models of how the kidney eliminates drugs on chip. And I will say that 30 of the 200 most prescribed drugs are eliminated by the kidney. And often it's a bit of a black box because if they're substrates for tubulus secretion, via basolateral, via the organic, anionic, and cationic transporters, it's very hard to predict in silico how drugs are going to be eliminated. But we have developed a 35-component compartment model for how the kidney eliminates drugs. And then we've tested drugs on chip and applied them to the model, and then applied that to existing in vitro PK data, and we can match it. So we can show that we can model in basically in vitro complex, and combining in vitro with in silico, we can model complex pharmacokinetics. And then the third area is drug discovery and drug development. And I'll show you two examples of the work that we've done in iPS-derived kidney organoids. Autosomal dominant polycystic kidney disease is one of the most common genetic diseases in the world that has an end organ phenotype causing end-stage kidney disease. 1 in 1,000 people worldwide inherit a mutated version of one of the two genes, polycystin 1, PKD1, PKD2, which encode for polycystin 1, polycystin 2. PKD1 looks like a G protein coupled receptor. PKD2 is a cationic channel. They form a complex in the cilia, and they form cis later on in life when they're mutated. And it's still not very well understood. There's only a single FDA approved drug, and it has a lot of side effects and is only modestly effective. So there's an unmet need for new drugs for PKD. And Bino Friedman in our group, Benjamin Friedman, 10 years ago now almost, made iPS cells into kidney organoids, used CRISPR-Cas9 to knock out either one of the two genes, created a cystogenesis phenotype in vitro. Nellie Cruz then found a way to increase the cystogenesis expression up to 60–70%, which allowed for high throughput screening with robotics. And we found new pathways in vitro that hadn't previously been examined as potential therapeutic targets. And we've done a lot of work since then using drugs that are in the clinic and drugs that may be getting into the clinic to show efficacy or lack of efficacy in cystogenesis now in vitro. The other disease entity that we proposed is cystinosis. Cystinosis is a rare disease. It's an autosomal recessive lysosomal storage disease, whereby the amino acid cysteine, which can form an oxidized homodimer called cysteine, accumulates in lysosomes. And it's a mutation of the transporter that lets cysteine out of the lysosome. So there's an accumulation of cysteine. It's a really terrible disease where the children develop end-stage kidney disease, usually before the age of 10, because of lysosomal dysfunction. They also develop crystals in the eye and photophobia, and can have loss of vision as well. There's only one known therapeutic at this point in time. It's a rare disease, which is modestly effective, cysteamine. So we made a model again with kidney organoids and CRISPR'd out the relevant gene, showed that we can make organoids. And yes, we get the same phenotype where you get cysteine accumulation in the organoids that no longer, that have basically knocked out the gene. And then we have shown that in addition to the phenotype, the one drug that is in the clinic, cysteamine, is effective in decreasing the cysteine accumulation in organoids. And we have a cell death phenotype in the organoids as well. So the second context of use from the drug discovery, drug development, from our perspective, is to treat a rare, really devastating autosomal recessive disease. Two other quick slides, and I'll end. As part of our consortium, the cell source, I think everybody in this room who is involved in microphysiological systems or complex in vitro models know cell source is critical, and we do have a company that we are working with that was started out of the University of Washington that has biobanked a number of different iPS cells that we can use for the work with different mutations. And then we've had a lot of industry support for our application. This was the industry support when we submitted our application, again, both biotech and large pharma. And we've had a number of other larger and small companies that have been talking to us over the past year or so as well. And I think that's, oh, here's the last slide, which is just the layout that we put in the grant for how we're going to go about the process of moving these into the drug development tool qualification process. So thank you for your attention.